All right, today we get to play chemical detective. We're diving into a study that starts out looking pretty simple, just a fluorescent molecule that starts to dim. But what it uncovers is this really surprising destructive force that was hiding in plain sight. It's a fantastic reminder that in science, what you see at first glance is almost never the whole picture. So here's how we're going to break down this mystery. We'll lay it all out in 5 parts. First, we'll meet the main characters, the molecules involved, and set the stage. Then we'll look at what seemed to be happening, a classic energy transfer story. After that, we'll follow the trail of clues that just didn't add up, uncover the real disruptive force at play, and finally, we'll put all the pieces together to see what was really going on. Okay, first things first. Every good mystery has its cast of characters, right? So let's start by getting to know the key molecules here and see how the whole experiment was set up from the beginning. So the star of our show is this fluorescent molecule, our donor. Its full name is a mouthful, dimethylaminocyanobiphenyl, so we're just going to call it DMACB. And how do they make it? Well, they use a really well-known chemical reaction called a Suzuki-Miyaura coupling. You can think of it like chemical matchmaking. It uses a palladium catalyst to basically snap 2 smaller pieces together, BRA-DMA and CNPBA, to build our final super bright DMACB molecule that we need for the experiment. Okay, so here's the thing about DMACB. It's hydrophobic. That's just a fancy way of saying it hates water, so it won't mix in. To get around this, the researchers used a really smart trick. They brought in a surfactant called CTAB. These CTAB molecules form these tiny little protective bubbles in water, which are called micelles. They basically create a cozy, water-free home for the DMACB molecules right in their center. This gives us these really stable, nicely organized, and super bright little bundles, which we'll call DMACBA-SS. And of course, for any good experiment, you need a control group. So they also made a version called DMACBA-R, which is just the DMACB dumped into pure water. No protective bubbles. All right, our stage is set. We've got our bright donor molecule all bundled up nicely. Now the researchers bring in the next character, an acceptor molecule called fluorescein. The goal was to see if it would quench or dim the light from our donor. And the initial hypothesis? It looked like a classic open and shut case of something called Forster resonance energy transfer, or FRET. And the first piece of evidence really comes down to what these things look like. Just check out these microscope images. On the left, that's our control sample in water. It's a mess, right? Just a chaotic jumble of plates. But look at the right. With that CTAB surfactant, you get these beautiful, perfectly formed little rods. This is huge. It tells us the surfactant isn't just keeping them from clumping randomly. It's actually creating this incredibly organized structure. And that organization is key. The spectroscopic data seemed to seal the deal too. See, for FRET to work, the light the donor gives off has to be the same kind of light the acceptor likes to absorb. And what do we see? A perfect match. The overlap is excellent. Plus, our CTAB-stabilized donor is seriously bright. We're talking a quantum yield of 66%. That makes it an awesome energy donor. Meanwhile, the control sample in plain water, super dim, only 3%. So at this point, everything is screaming FRET. It looks like the perfect setup. But you knew there was a but coming, right? This is where our simple story takes a hard left turn. When the scientists zoomed in on the data, they started noticing some really weird things, anomalies that just didn't fit the simple FRET story. Okay, so they add the fluorescein, and just like they thought, the donor's light starts to dim. That's quenching, that's what we expected. But when they looked at the color of the light, things got strange. The donor's light shifted towards blue, a higher energy, while the acceptor's light shifted towards red, a lower energy. Now think about FRET. It's supposed to be a hands-off process. It's like using your voice to make a wine glass vibrate from across the room. You don't actually touch the glass, so you don't change the note it rings at. FRET shouldn't change the note or the color of either molecule. So these shifts, that was the first huge red flag. Something else had to be happening here. And then came the time resolve data, which is where things got really interesting. They measured how long the donor molecule stays lit up, its lifetime. Normally, it was 0.17 nanoseconds. But with the acceptor there, it plummeted to just 0.03 nanoseconds. Okay, dynamic quenching, got it. But here's the kicker, the absolute crucial clue. Remember our messy, dim control sample in pure water? Its lifetime was 0.02 nanoseconds. That's basically the same number, 0.03 and 0.02. That cannot be a coincidence. It's as if the donor, once it was quenched, was somehow turning into the control sample. So, with all these weird clues piling up, the researchers had to rethink everything. They started to wonder, what if the acceptor molecule isn't just a passive bystander? What if it's not just accepting energy, but it's actually attacking the system? And folks, this right here is the smoking gun. On the left, you see those nice, neat rods we started with. Now look at the right. After adding the fluorescein, they're gone, completely obliterated and replaced with that chaotic cluster. The acceptor didn't just dim the donor, it physically wrecked its home. And the data confirms it. The disruptive effect was way worse when there was less surfactant around, where the micelles were weaker. Suddenly, it all made sense. Fluorescein is negatively charged. The CTAB surfactant is positively charged. Opposites attract. The fluorescent was literally ripping the protective micelles apart with electrostatic force. Okay, so now we have all the puzzle pieces, and we can finally see that this was never just a simple story about fret. No, it's actually a tale of two different things happening at the exact same time. This final diagram sums it up perfectly. You've got two pathways going on. Pathway 1 is your standard fret. For any micelles that are still intact, yeah, they're transferring energy to the fluorescent, just like we thought. But at the very same time, you've got pathway 2, the destructive path. The fluorescent is attacking the CTAB, breaking open those protective bubbles. And when they break, the DMACB molecules inside, which hate water, are suddenly exposed. They panic, and they clump together into those dim, disorganized aggregates, just like our control sample. And that is what explains everything. The blue color shift, the huge drop in lifetime, it all clicks into place. So what's the big lesson here? Well, as the authors put it, it's the need for a deeper analysis to avoid jumping to easy conclusions. The dimming they saw wasn't just fret. It was a complex dance between fret and this destructive electrostatic attack. And that's actually a pretty big deal, because it shows the acceptor molecule wasn't just a passive observer, it was an active participant that was changing the entire experiment from the inside out. It's a critical reminder for any scientist, really. You always have to ask yourself, is my measurement tool accidentally breaking the very thing I'm trying to measure? It's a question that keeps science honest and pushes us to get the real story.